My lab actually focus on the study the brain development. I would like to actually majority one of the interesting we are focused on is how the brain gets larger. So you can see one of the most remarkable feature of the evolution of the brain is the enormous increase in the number of neurons, which reached the peak in the human brain. And also, although the lamina structure, they share a similar lamina structure in all mammals, the alteration of the cortical surface actually underscores the importance of you can see from the mouse smooth brain to the gyrocephalic human brain, which underlies the importance to understand how to generate such a complicated structure, which underlies to understanding the process of neurogenesis during the brain development. I would like to bring your attention for this movie. You can see from this movie, you can see here, actually you can see this is we take the time-lapse imaging from the lateral ventricle of the mouse. You can see you can see here along the lateral ventricle, there are dynamic interconnected nuclear membrane of the radial glial cell actually located within the ventricular zone we call VZ in short. And also there are some cells actually divided symmetrically above the ventricular zone located in the we call subventricular zone in short, called in short in the SVZ. The cell actually divided locally, just symmetrically divided. So this is generally the point that we in the field. The point of the field actually there expanded the neurocortex in the pyramid. Actually, we focus on two cell types, like the A cell type, which we call the neuroepithelial-like cell, or the radial glial-like cell, radial glial cell, and also another cell we call B cell, which is we call the intermediate product cell, or the transit amplified cell, which symmetrically divide and generate two neurons. And this cell actually usually generates one neuron, but the cell actually located within the ventricular zone, and this cell located above the ventricular zone in the subventricular zone, so they can sort of like divide in this area. If we think about how to expand the proliferation capacity, there are two ways actually. We can expansion of the founder population. We can get a maybe larger ventricular zone like this, and also we can increase the transit amplify. We can get the larger subventricular zone here. The cell actually just like this. Usually they just divide locally from 1 to 2. Yeah. But if we look at the human fetal brain, actually we get a generous donation with constant letter. We can see the dramatically expansion in the above the subventricular zone. You can see the dramatic expansion we call OSVZ in short, actually full of the progenitor cell. You can see relatively the ventricular zone and the subventricular zone is not like we proposed the model. You can get large expansion. So if we look at it carefully, you stain with a different marker like a SOX2, stain the radial glial cell or progenitor cell, or the TBR2 we stain with the intermediate product cell, stain with this green, show with the green color. You can see from the human actually in the VZ and also subventricular zone share the similar pattern, and then not like expansion dramatically. But in the OSVZ, you can see dramatic expansion in this area full of the SOX2 positive cell and also TBR2 positive cell, which might suggest in the human, actually, the expansion really have a different progenitor, that might have a different progenitor to contribute this dramatic expansion. So if we look at carefully in the single cell resolution, you can see along the mouse ventricle, there are just like a radial glial-like cell, which also looks like an epithelial cell. If they get elongated and all the fiber reach expansion to the pair surface, the cell actually can divide, generate the cell, migrate along the among the radial cell all the way to the pair to form form the cortex. How about the cell located in this area? So back to 10 years ago, maybe 12 years ago, actually in the Arnold Crystal lab, they actually perform the time-lapse imaging. You can see. The cell actually located here, and they actually are not divided interkinetically, but actually just the shooting up and divide. So sort of like the cell actually can jump up and divide. We call mitotic somal translocation. They generate self-renew OSVZ product cell, or generate another cell which we call the intermediate product cell. The cell continue divide symmetrically to generate two daughter intermediate product cell. Actually, we perform the time-lapse imaging for a couple of days, and we never see the cell can directly generate neuron, but instead actually generate a whole bunch of like intermediate product cell. So that's the reason they can get an expanded OSVZ region. So that's my sort of serve as a clue for how the dramatic expansion in the OSVZ region. So we actually proposed actually if we have another subtype like a neuron product cell located in above the subventricular zone, different with the radial glial cell limited within the ventricular zone. Actually, the cell can jump up, divide, might generate a lot of daughter cell which can continue divide. Eventually, they will generate a whole bunch of the neuron. That's the whole hypothesis for what we try to understand the gyrocephalic brain formation. So if we look at another like a species like a ferret, which have a gyrocephalic brain, and actually if we look at this sort of like have a similar OSVZ region full of like frontal cell can divide, jump and divide, but also have a different pattern of the division, sort of like they will generate neuron directly or indirectly. And actually, if we knock out some gene related to the gyrocephalic formation, actually, you can see we can dramatically reduce the OSVZ product cell and also get a very smooth brain. So that suggests that this OSVZ product cell actually might have the secret or potential to generate the expanded brain and also the gyrocephalic brain. So we proposed with this model, like in the neuroepithelial cell in the human, they might just elongate it to turn to the radial glial cell, and the radial glial cell might symmetrically generate one neuron with low efficiency. Later, if they generate the ORG or OSVZ product cell, they might can have a fully potential to get multiple duplicate and also generate a whole bunch of neurons to contribute to the gyrocephalic brain. So how to validate this hypothesis? So actually we get the generous donation from the consent letter. We collect the early embryo, like from the Carnegie stage 10 to the Carnegie stage 12. Actually, we do the whole bunch of the single cell iron-seq. Actually, this work was inspiring by with Fu Chou Tang and also Sunny. So actually, you can see we can get a whole bunch of the cell subtype, and also we can get 11 different lineages from like you can see here from like neural tube, neural crest, and we might can figure out where are the earliest neurogenesis from. So you can eventually we can get actually you can see the cranial, the caudal origin of this from the cell, and also we can see the diversity of the radial glial cell from here, origin from the neuroepithelial cell to generate a different type of the subtype, and also the different subtype of radial glial cell have a directly lineage to different subtypes of the neuron from very early stage. Actually, you can see, and also we figure out the neuroepithelial cell marker, how they transform to the radial glial cell, and then generate different type of the neuron located in the different part of the brain and also spinal cord. We also can get with the spatial transcriptome, we can map and the different neuroepithelial cell marker and neuroepithelial cell type, and also radial cell type back to the different part of the embryo. We can see actually clearly, you can see different subtypes like radial glial cell located in the forebrain, like hindbrain, and also spinal cord. So that, which suggests the different subtypes of the radial glial cell might have their location information, which contribute to different part of the neuron generated in different part of the brain and also spinal cord. We actually also figure out during the neuroepithelial cell turn to be the radial glial cell, they need like an epithelial mesenchymal transition actually, and also need a lot of like coherent protein like ZO1 and CDH2 to be adjusted to be turned down, and also to get into the radial glial cell type. Although the conversion of sort of like from epiblast cell into the neuroepithelial and their subsequent transformation in the radial glial cell is actually very critical. And from this process, we also identified the OSVZ-like cell, which is marked with the Hopax positive cell, actually can show up in the very early stage, like a PCW, just in week like a 9, which suggests we can from this data set we can pull out all of the information from the neuroepithelial cell, transform to the radial glial cell subtype, and also transform to the OSVZ progenitor cell. So actually, we are dying to know what exactly OSVZ progenitor cell, the difference OSVZ progenitor cell to the radial glial cell or to the neuroepithelial, even to the neuroepithelial cell. So collaborate with Fu Chou Tang a couple of years ago. We actually tried to use a single cell iron-seq data technique to pull out the OSVZ region, and we use FACS sorting to sorting out the different cell type, which actually we verified with real-time PCR. We can actually get ventral radial glial cell, OSVZ radial glial-like cell, and the intermediate process, and also neuron. Then we can perform the very deep sequencing, and actually we identified a whole bunch of genes enriched in the OSVZ radial glial-like cell, or org cell in short. So one of the genes actually, which we call the TMEM14B, which is a transmembrane protein, actually with non-function there at that time, and actually also the primate specific gene. So how to validate the gene's function related to the OSVZ region? One of the things actually we put this gene overexpresses this primate-like gene back to the mouse brain. So actually, if we overexpress this gene, we make a transgenic conditional knock-in mice. Actually, we can boost the forebrain of the mouse to get a gyrocephalic-like folding structure, which suggests this one gene sort of like can help and contribute to generate more cell, which is actually leading to this SVZ region generate more ORG-like cell. Actually, they might can generate more neuron to boost the cortex to get a folding structure. And we also identified the IQ gap1, which is RAS GTP activating-like protein associated with the TMEM40B. Actually, the major function is like they can boost the G1 to S transition, which is to promoting the G1 to S phase transition to boost the generate more OSVZ-like cell. Actually, we can get the gyrocephalic-like structure. Then how about the intermediate progenitor cell in the human? It is known that in the human, the cortical intermediate progenitor cell could divide a million neurons to expand the progenitor pool. which was absent in mice. So in the mice, usually intermediate process only divide 1 time or 2 times. When we investigate the single-cell transcriptome profile for human developing a cortex, which will actually also collaborate with Fu Zhou-Tang, which was mainly done by Sui Jun and also Xiaoqun from Fu Chou's lab. We found that the developing trajectory of neural protein cell follows 3 major patterns here. And when it actually start from the with a different start point. And also, we actually conclude that when the early path is from radial glial cell to the intermediate cell. Also, second route is radial glial cell to the ORG cell. And also third route is actually from radial glial cell to ORG cell to RPC cell, which actually can validate by immune staining the early stage. You only can see the ventricular zone full of radial glial cell and sort of a little bit intermediate cell, but in the later, like just in week 16, you can see a whole bunch of like radial glial cell limited here, but the whole of the expansion of the OSVZ region full of intermediate cell, which actually were generated from ORG-like cell. So we draw the conclusion we can, in the human prefrontal cortex development, actually they have a two-peak neurogenesis, which will drive dramatically by early stage intermediate cell and also later stage intermediate cell. And both actually we use ion in situ hybridization to validate this candidate. Actually, you can see the whole bunch of the intermediate cell. Different genes actually have a different expression pattern, which also suggests the intermediate cell might have a different subtype, which we probably need to follow up to know how they contribute to how the pattern of the division, what kind of daughter cell they generate contribute to the cortical formation and also gyro formation. If we actually, other than the ORG cell and also intermediate cell, there are another species we call Chinese tree shoe. If you look at the Chinese tree shoe brain, they have no gyrocephalic structure, smooth brain, but the brain can expand it. But from the expansion neural cortex, we could only identify a majority cell actually from intermediate cell, which might suggest during the evolution, the ORG cell come out a little bit later. Before that, actually, the majority expansion really depend on the different subtype of intermediate cell, which contribute to the expansion of the tree shoes expanded the brain. Meanwhile, if the cortical interneuron actually were generated from the ventral part of the we call MG or LG, which represents the ganglion eminence, which is the medial and also lateral ganglion eminence. We look at the whole brain interneuron origin and particularly focus on the MG and also LG potential. We can identify actually cluster 19 human-specific MG protein cell. Actually, which suggests there are some human-specific MG protein cell actually might generate more interneuron, which migrate to different parts of the brain, scale up, because the excited neuron and the inhibitor neuron might have a relatively stable ratio to maintain the excitatory and the inhibitory balance. So for the human actually development, the MG part might also have some specific unique protein cell which generate more interneuron to proportionally to match the excited neuron expansion. Other than the lateral ventricle and also ventral part, if we look at the third ventricle, which most of the radial glial-like cell generated cell actually contribute to the hypothalamus. If we look at use some dye to backfill from the pear surface, actually you can see we can also pull out some cell with monopolar shape without the basal. We only have the basal fiber without the apical fiber anchored in the third ventricle, which might suggest that they might have a similar ORG-like, OSVZ-like cell in the hypothalamus. If we do the time-lapse imaging, we also can see the similar mitotic somal transcription pattern, which suggests even in the human development from the third ventricle, which generally majority of neurons contribute to the different nuclear area of the hypothalamus, they must also have a similar neurogenesis pattern or neurogenesis different subtype of neurogenesis function for the scale up the proportionally scale up to the hypothalamus region. If we look at it carefully from the along the developmental stage, we actually can pull out all of these. We can actually pull out a lot of progenitor cell contribute to the contribute to the hypothalamus area, which is actually full of almost 25 different subtypes, and actually they exhibit significant lineage differences across the human hypothalamus development process. To gain the insight, we actually we perform a regular analysis use a specific algorithm, which actually can see how the by transcription factor contribute to the to divide and contribute to different subtype development, which actually we can pull out the transcription factor regular how to generate different subtypes of hypothalamus neuron. And to define gene expression pattern in human hypothalamus development, actually we adapt a transfer linear approach to target mouse cell type, because in the mouse cell type they are actually already defined very well how many subclasses of the cell in the hypothalamus. But actually we use a transfer linear to map the human cell type to the mouse cell type. We can get a different pattern. One of the we can from all of this among all of this pattern, actually we can find some pattern specifically belong to the human hypothalamus. And also actually according to the correlation between the pattern feature and also major cell type in the human and the mouse data set, we define strongly associated pattern with each cell type. And we also found some cell actually specific, some gene actually during the development actually specific expressed in the human-specific gene expressed in the hypothalamus. Further, actually, we also detailed analysis of neuron progenitor cell highlight the unique developmental feature in the trajectory of the neuron progenitor cell for hippocampus, which is related to the linear memory for the human brain. Actually, we also proportionally found some human-specific gene related to the specific subtype of neuron progenitor cell for hippocampus. If we boost an overexpressed gene in the mouse, actually we can get a larger hypothalamus structure, which suggests the gene human-specific gene might also contribute to scale up the hippocampus for the linear for the high cognitive function of the human brain. Because they are in the field whether adult hippocampus neurogenesis persisting in adult, so we sort of use a single cell iron-seq single nuclear iron-seq technique. Actually, we can pull out the continue the newborn neuron and also proliferating adult neuron progenitor cell in the very aged human hippocampus, which might suggest actually the existing of immature neuron and also continue neurogenesis in the aged human brain, which is actually solved the debate in the field. I collaborate with a doctor and also collaborate with Professor Zilong Qiu in Shanghai. Actually, we try to apply our single cell data with autism disease cohort. Actually, we can map the cell type and also map the brain-specific region to the specific neuron progenitor cell subtype. We actually correlated with the low function or high function autism disease, which actually suggests with single cell iron-seq data, even within the during the developmental state, actually you can also contribute to the disease study. Actually, we have another example for disease study, which sort of like a disease which have microcephaly, which has a neuronal progenitor related disease, which have a smooth brain, or lissencephaly, which have a smooth brain, or primate macrocephaly actually have a complicated gyrocephalic structure. Also, they also have some like a macrocephaly with a very less gyrocephalic. So one of the genes actually relate to the centrosome. If we knock down the gene, knock down this gene, actually CPAP-G relate to the centrosome elongation. Actually, you can get a small brain, and also completely loss of like a lot of neural progenitor during the mouse development. Do something like experiment with the invasive like experiment. We use like we actually build up or set up the vascular structure with the organoid. Actually, we can generate not only the radial glial cell in the dish, but also intermediate cell in the dish, and also we can generate the ORD-like cell in the dish. Actually, can perfectly mimic 95% of cell, include the neural progenitor cell, can mimic the human fetal brain development. If we transplant the human organoid, actually they can continue grow in the mouse brain. Actually, can generate sort of like a gyrocephalic structure, and within the vascular like the organoid, you can see they have a blood that actually can penetrate, go through the blood vessel within this organoid transplant into the mouse brain. So with that, actually we can sort of like mimic the macrocephaly in dish, and we can actually generate, we can get a patient RPS cell and generate the macrocephaly organoid, and actually you can see we can get mimic the neurogenic pattern, and also we can get like a less, or sort of like a electro physiology data actually also suggests that when they get a groove and in a mature neuron, they can have a less like a neuron circuit or neuron connectivity, which can be revealed by the EPSP or RPSC data. So, I would like to stop here and summarize with the single cell, the powerful data. Where eventually we can get access to the human tissue. Then we can actually review the very spatial information of the radial glial cell in early embryonic development, and also we identify or really know how the neuroepithelial cell transform to the radial glial cell, and also by how what kind of the signal pathway relate to this process. And also we can decoding the diversity and the molecular characteristic of the neural progenitor cell, like RT cell, ORT cell, and RPC cell, and also all related cell lineage in the developing human brain in the different region. And also can illustrate the cell type and the gene expression evolution of human brain progenitor, which actually can help us understand how the brain get bigger, and also how the brain get the high function for us to be a human. I would like to stop here. Thank you.
All right, thank you.